Okay, so today we're going to look at anaerobic respiration. This has two steps. The first step is glycolysis, which is identical to the one in aerobic respiration. And second, we have fermentation. There's a difference. You can have alcoholic fermentation in plants and yeast, and lactate fermentation in bacteria and some animal cells. So firstly, let's begin with glycolysis. Glycolysis happens in the cytoplasm of all cells, so therefore it can be used in both anaerobic and aerobic, as it does not happen in the mitochondria. Glucose is firstly phosphorylated using ATP to form glucose phosphate. Glucose phosphate is phosphorylated again to form hexose bisphosphate. Hexose bisphosphate then splits into two molecules of triose phosphate. Triose phosphate is then oxidized, meaning it loses hydrogen, to form two molecules of pyruvate. As this happens, NAD also collects the hydrogen ions, forming two reduced NAD. Four ATPs are also produced in each step. So two are formed with one conversion of triose phosphate and two with the other. So now if you look at the number of carbons being recycled throughout this process, we started off with glucose, which was six carbon molecules. However, when it converted into triose phosphate, it converts into three carbons, and therefore pyruvate has three carbons. Next step involves fermentation. We have two forms, alcoholic or lactate fermentation. So let's begin with lactate fermentation. Lactate fermentation occurs in animal cells and in some bacteria. Pyruvate is directly converted into lactate, or also known as lactic acid. As this happens, NAD is regenerated, so NADH converts back into NAD, and this then goes back into glycolysis. This enables glycolysis to continue to occur so that we don't always need oxygen for the production of ATP. In alcoholic fermentation, pyruvate is firstly converted into ethanal, with the removal of CO2. And then ethanal is converted into ethanol, and this process also regenerates NAD. NAD is also fed back into glycolysis.